Hello everyone, and welcome to today's presentation titled A Novel Synthetic Self-Amplifying RNA System and LNP Delivery. This webinar is a part of the 4th annual event in the Immuno-Oncology Virtual Event Series. I am Melanie Chen of LabRoots, and I'll be our moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now, let's get started. I now present today's speaker, Kangming Chen, Head of mRNA and Plasmid Department at GenScript. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Kevin, you may now begin your presentation.
Thank you for your time to come to my presentation today. My name is Kangming Chen. I'm the head of mRNA and Plasmid Department in GenScript. Today, I'm going to talk about a novel synthetic self-amplifying RNA system and LNP delivery system developed by GenScript. So my talk will mainly cover 4 different aspects. The first part is a quick background introduction, and then about GenScript self-amplifying RNA and LNP platform introduction. And then I will introduce a little bit about our self-amplifying service. And in the last part, I will introduce the GenScript peptide service and products. So here is vaccine development process, and GenScript can support you from early research to clinical trial. And at the very beginning, scientists usually need to identify or select an antigen for vaccine development. So during this process, GenScript can help you on pathogen study like genome sequencing, antigen identification and selection, or antigen epitope study. Specifically, we can use neoantigen peptide synthesis to help the tumor study. After the scientists identify the antigen, they need to do a vaccine sequence design, modification, and preparations. So during this process, GenScript can provide like gene synthesis and plasmid preparations for DNA vaccine development, and mRNA synthesis and LNP for mRNA vaccine, and mammalian CHO expression for recombinant protein vaccines, and the neoantigen peptide synthesis for peptide vaccine. So then the project will move into preclinical study for vaccine effector evaluations. During this process, GenScript can provide antigen expression test, humoral immunity evaluation, and cellular immunity evaluations. So even the projects move into clinical trial, GenScript can provide a GMP-grade plasmid DNA, peptide, or biologics. And we can also provide some custom ELISA kit for host cell protein, neutralization, etc., and also like endotoxin detection kit, etc. So here is a roadmap for the tumor neoantigen development. So scientists has realized the importance of immunology many years ago, back to the year 1943 and the year 1988, scientists found a new type of peptide in mice, excuse me, and called it a new antigen peptide. This is the first time for establish of the concept of new antigen peptide. Then in 2017, two papers published in Nature caused a lot of attention in academic field and put the new antigen peptide study to the next level. So, what is new antigen peptide? Why it play a key role in immunology treatment and cancer therapy? So, new antigen peptide is a tumor specific. It only exists in cancer cells. It can be produced by virus infection or gene recombinations, and it can be delivered to the T cells by antigen presenting cells to activate the immune system. This kind of abnormal peptide we call it new antigen peptide. Their exclusive expression in tumor cells makes them highly suitable and safe target for cancer immunotherapy, and it's opened a new chapter for cancer treatment. So now I'm going to introduce a little about the mRNA vaccine. So here is a picture shows the typical process for mRNA in cells. The mRNA was produced in the nucleus and was exported to the cytoplasm. In the cytoplasm, it can binding by the translation complex. So there's many protein involved in this process, such as a polybinding protein, and eventually it will form a loop and then turn on the translation. So here is a picture shows the translation process. This line is the mRNA template, and this little black dot is the ribosome, and this branch structure is the nascent protein or peptide. Another fate for the mRNA in cell is degradation. So the mRNA can be recognized by different enzymes from the 5' or the 3', and eventually it will be completely degraded by these enzymes. So the half-life of mRNA is very short. It can only last about 1 or 2 days. So today I'm going to show something about the self-amplifying RNA. So here is a typical process for the linear mRNA, and this is the process for the self-amplifying RNA. So the self-amplifying RNA contains non-structured protein 1, 2, 3, 4 from the alpha virus. It can form replicones, and these replicones can specifically bind into the subgenomic promoters and replicate the downstream target genes and make multiple copy of target genes. And this gene will be used as a template to produce the proteins. So here is a picture shows the lipid nanoparticle structures. It has multiple lipid components, such as the positive charged ionizable lipid and phospholipid and cholesterol and the PEC lipid. All this lipid can together with the mRNA will form a round shape and a stable structure, which we call the lipid nanoparticle. So the self-amplifying RNA has been used for COVID-19 vaccine development. In 2023, Japan approved the first COVID-19 mRNA vaccine based on the self-amplifying RNA strategy. Excuse me. And earlier this year, this self-amplifying RNA vaccine was approved in European markets. So the self-amplifying RNA has been used for many infectious disease vaccine development, such as RSV, influenza, coronavirus, etc. It can also be used for cancer vaccine development targeting the HER2 or the neoantigen vaccine development. So in the second part, I'm going to introduce GenScript self-amplifying RNA and the lipid nanoparticle platform. So previous study has shown that the VEV virus was widely used in vaccine development. So in our lab, we want to find a different self-amplifying vector with higher protein expression, and also because the self-amplifying RNA was from the alpha virus, and as we know, the alpha virus can target different tissues or organs in animal or in humans. So we also want to identify a different self-amplifying RNA vector has tissue-specific targeting capabilities. Follow these two ideas. We we did a screening. So what we did is first we synthesized 30 different self-amplifying RNA vectors, and we used the reporter genes to replace the structure proteins in the designs. And then we we check their protein expression in vitro cell functional assay, and also validate the tissue-specific targeting in vivo mouse experiment. So totally, we identified the 3 self-amplifying RNA with higher protein expression and also with tissue targeting capabilities. So here is in vitro cell assay data. So we have the VEV self-amplifying vector as a control here, and we can see many different self-amplifying vectors showed higher protein expression in our cell assay experiment. So after we identify the 3 different self-amplifying RNA vector, firstly, we try to compare the transfection efficiency for lipofectamine and the lipid nanoparticle. And as you can see, the lipid nanoparticle transfection strategy showed much higher EGFP protein expression in the experiment. And then, as we know, the linear MI, and as we know that the linear MI requires modification to the uridine. So we also want to know whether or not self-amplifying RNA requires any modifications. So we made a different modification sequence versions like M5C modifications, pseudouridine modifications, and one methyl pseudouridine modifications. And we test this sequence in two different cell lines, HEC29T cells and A549 cells. And you can see the M5C modifications showed much higher protein expression in both cell lines. And we sent this M5C modified self-amplifying RNA sample to our cooperator labs in Singapore Nanyang Technology University. And you can see the M5C modified version showed much higher protein luciferase expression than the unmodified SNR vector sequence. And as we know, the linear MI requires quite long poly tails. So we want to characterize whether or not self-amplifying requires a long poly tails or not. So we made 40As, 70As, and 100As in the sequence, and then do the protein expression test. However, we did not observe a significant difference between these three different poly-A designs. So by default, we always use like 40As or 70As in our designs. And then we try to characterize the dosing for the self-amplifying RNA, because the self-amplifying RNA can replicate the target genes, and which means it can lower the dosing in the treatment. So in this experiment, we use LNP to deliver the self-amplifying into three different type of cell lines: HEC293 T cell, A549, and Jacquard cell lines. And we load 10 nanograms self-amplifying into the cells, and as a control, we load 100 nanograms for linear mRNA and 100 nanograms for circular RNA. And you can see in this experiment, even though we know the dosing for the self-amplifying to 10 nanograms, it still shows comparable protein expression compared with linear mRNA and circular RNA. In some cases, it showed even higher protein expression than the linear and circular RNA. Because self-amplifying RNA contains some sequence from the alpha virus genome, so usually it can cause higher immunogenicity than the linear mRNA and the circular RNA. So in this experiment, we try to characterize the immunogenicity for three different types of self-amplifying. So we measure the immune markers expression levels in the samples, for example, IL-6, interferon alpha, and IP10. So as you can see, the self-amplifying -4, indeed, it shows higher immunogenicity in IL-6 and interferon alpha, but not in the IP10. And for the self-amplifying -1 and -3, they showed quite low immunogenicity or comparable immunogenicity with the linear mRNA and the circular RNA. We also quantified the mRNA transcription levels for these three different types of RNA. excuse me, we note 100 nanograms linear mRNA and 100 nanograms circular RNA and 100 nanograms self-amplifying, and we can observe much higher EGFR levels in A549 cells at a different time point. So after we done this in vitro cell assay experiment, we also want to know their protein expression patterns and biodistributions in vivo mouse models. So we inject 3 different self-amplifying into the mouse, and the linear mRNA was used as a control here, and also the negative control injected with PBS only. So you can see the linear mRNA showed significantly higher luciferase expression at day 1, day 2, and then it's quickly degraded after 1 week. And then for the self-amplifying, we can observe the luciferase expression even after 3 weeks later. So in this figure, you can we quantify the luciferase expression signals. Again, the linear mRNA shows very high luciferase expression at day 1, day 2, and then quickly going down. And the self-amplifying shows very stable protein luciferase expression in the mouse, and it lasts for more than 3 weeks. So, as I mentioned, we also want to identify different self-amplifying vector with capability targeting different tissues or organs. So linear mRNA was used as a control here. It's not surprised that it's mainly enriched in the liver, but for the 3 different type of self-amplifying vector, we are very excited to see that they show different biodistribution patterns. For example, SLN-1, the luciferase expression was mainly observed in heart, spleen, and kidney. And for SLN-3, luciferase expression was mainly observed in spleen and the lymph node. And for SLN-4, the luciferase expression was mainly observed in spleen and kidney. So, and then we want to know was this tissue specific targeting was caused by the LNP delivery system. So, to answer this question, we labeled the SM1 and 2 with a DIR fluorescence dye, and then we checked these DIR fluorescence signals from these different tissues or organs. And surprisingly, that all the DIR signal was mainly enriched in the liver. So, which indicated that the LNP delivery system did not cause this different biodistribution patterns. So, after we established our self-amplifying RNA platform and our LNP delivery system, we tested this technology in GPCR transmembrane antibody development projects. Because previously we tried to use purified antigen or plasmid DNA to immunize the animal, the mouse or the rabbit. We did not get any positive clones. So in this case, we make the self-amplifying contain the CCR9 genes and encapsulated in SM102 lipid formulations, and then inject into the rabbit. And as you can see, after the second immunizations, we can observe positive signal in the serums. So totally, we screened about 400 positive clones using the FACS method, and then we selected top 20 clones validated by our NGS sequencing, and then we produced 10 recombinant antibody and validate in our FACS EC15, and then 9 of them shows really good performance. In another case study, we use self-amplifying to express the COVID-19 RBD protein, and this self-amplifying was encapsulated in SM102 LNP formulation, and it's directly injected into the mouse without adjuvant. So you can see after we immunize the mouse, we collect the serum, we check the serum titles by ELISA at different time point. Even after 3 months, we can detect very strong serum antibody titles. So, in summary, GenScript developed a novel self-amplifying vector -1, -3, -4. They shows much higher protein expression than the traditional VEV vector, even with much lower dosing. And the majority of siRNA delivered by the MP using SM102 formulation enriched in the liver, and the different siRNA vector has variable tissue targeting specific capabilities. So GenScript self-amplifying and MP platform was demonstrated in GPCR and the COVID-19 antibody development with a title detected at least 3 months. So now I'm going to quickly introduce our self-amplifying service. So we can provide different scales like micro, mini, midi, maxi, and different cap options, cap AU, cap AG, and also different purifications like the RU grade purification and preclinical grade purification using the Oliver DT, HPLC, and the different QC test. As I mentioned, we identified 3 different self-amplifying vectors, -1, -3, -4. They have been shown different capabilities in our experiment. So the -1 vector can be expressed in the spleen heart, and -3 can be expressed in kidney and spleens, and -4 can be expressed in the spleens. So you can choose the best vector based on your downstream application. And here is a quick introduction about the LNP service. So we can provide up to 100 mg of mi encapsulation, and the length can be up to 20 kilobase pairs, and it can be injected through IM or IV, and we can provide different ionizable lipid such as MC3, SM102, ALC0315, LP01, and we can provide different QC tests like the encapsulation efficiency, the size, the PDI, the theta potential, and the endotoxin detection. So in the last part, I want to introduce our new antigen peptide synthesis service. So as I mentioned in the very beginning, new antigen are usually expressed on the cell surface and caused by single amino acid deletion, insertion, or substitution. So this makes them very difficult to synthesize and purify. In GenScript, we can our new antigen peptide synthesis can cover different lengths, such as 8 to 50 amino acid in length, and even some charged amino acid and some hydrophobic amino acid. We can provide the new antigen in a very fast turnaround time, and even the small scales is available, and we can purify some difficult sequence due to its lower solubility and aggregations. So in 2023, scientists from biotech and genotech published one paper about the personalized RNA neoantigen vaccines in Nature. So they introduced about the autogene cevumeran. So autogene cevumeran is an investigational mRNA vaccine co-developed by biotech and genotech as an adjuvant therapy in cancer treatment to prevent cancer recurrency following the surgery. Autogene cevumeran is a type of a personalized neoantigen specific immune therapy containing different mRNA species that together can encode up to 20 neoantigens that have been identified in the tumors of the patients. The aim of the vaccine is to stimulate the immune system to mount an adaptive response to any remaining cancer cells with these mutations. So in the last part, I want to introduce our catalog peptide. So GenScript is continuously expanding and updating our catalog peptide products. And this peptide product can be used for cell treating cancer treatment or like cosmetic applications or KRAS study or OVA study. And we can even provide peptide libraries for our customers. And this catalog peptide has very competitive pricing and a very quick turnaround time. The stock are available worldwide from US, Netherlands, Singapore, and China. And in GenScript, we promise to provide a stable quality of peptide to our customers. All these peptide, catalog peptide, are purified by HPLC. So it's very reliable and has higher quality. So that's all the information that I want to share with you today. I'm very happy to take any question from you. Thank you.
Thank you, Kevin, for your informative presentation. We will now start the Q&A portion of the webinar. If you have questions you'd like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. Let's get started. Our first question asks, in the in vivo animal study, for how long can the expression of F-Luc be detected when using the saRNA?
Well, thank you, Melanie. That's a good question. So as I showed in my slide, the luciferase we inject in the mouse can last over like 3 weeks, more than 21 days. And so it's much longer than the NleB. And we have also done another test. So we inject the RBD domains, which is kind of like a COVID-19 vaccine, into the mouse, and we check the serum titers for over 3 months. So after that time point, we can still detect very strong serum titers in the samples. So I would say the saRNA can last much longer protein expression than the traditional linear mRNA. Yes, thank you, Melanie.
Awesome, thank you. Our next question asks, what is the underlying mechanism by which the delivery of saRNA exhibits a different biodistribution pattern compared to that of mRNA?
Well, that's also a great question. So right now we don't know the exact mechanism behind these different biodistribution patterns, but our hypothesis is that because the self-amplifying is original from the alpha virus, and different alpha virus can target different organ or tissues in the like humans or animals. So using this different self-amplifying vector, we guess that this SNR vector can also target different tissue and cells in mouse experiment. And also our data confirm these conclusions. So our hypothesis is that this different biodistribution was caused by the different capability of replicon from the self-amplifying, because like in different host cells, the replicon might have different interaction with the host cells, and this will cause different replication of the target genes in the cells, and eventually will result a different protein expression levels in the organs or in the tissues. That's the conclusion we have right now. Yeah, thank you.
Thank you, Kevin. The next question we received asks, how does the M5C modification help the F-Luc expression in vitro cell assay?
Well, this is a good question. So as we know, the self-amplifying RNA replicates itself at the first round of replications, and then after that, the first replications, the template does not have any M5C modifications. So what caused this different F-Luc expression in vitro cell assay? Our hypothesis is still like the template modification result from the different capability of the replicons. So the M5C modification can increase the MI half-life in the cells, and eventually will accumulate more replicons in the cells, and this replicon will replicate more target genes or target sequence in the cells and produce more proteins. So that's the conclusion we have right now.
Okay, great. And it does look like we have time for one last question, and it is, in addition to F-Luke, were there any other transgenes that had been tested using the saRNA?
Yes, except to the F-Luke reporter genes, we have done many other uh, tests like the Cas9 MI, which can be used in genome editings, and the EGFP fluorescence reporter genes can be used to quantify the gene expression. So we have done the test using many other type of genes in our experiment. So uh, our saRNA platform is it's kind of universal tools for other type of genes, and uh, it has a much longer half-life and accumulate more protein expression. So it's ideal tools to study the mRNA vaccines in your projects.
Okay, great. Thank you, Kevin. Do you have any final comments for our audience?
Yeah, thank you, Manani. And so as I talked in the slide and also answer the questions in the Q&A parts, the self-amplifying RNA platform developed by GenScript has shown much better protein expression and much longer half-life than the traditional linear mRNA. So it will be a very good strategy to study the mRNA vaccines for your target genes. And I'm very happy to work with any lab or groups that has interest uh, in this kind of applications. If you have any questions, please shoot email to me or contact me. So I will be very happy to work with you for your project. Thank you.
Thank you again, Kevin Chen, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for your interesting questions. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Please visit GenScript's booth in the exhibit hall after the webinar for more information and resources. Questions we did not have for time today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 30th, 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.